Thanks for joining us today at Right to Life of Northeast Indiana for our discussion about the COVID vaccines with Dr. Scott Myers. Very grateful to him for coming in and doing this live with us so that you guys can get a little more than what the media tends to share, some background here. And our hope is that this is helpful to you and that you'd be able to share this with friends who are considering the vaccines or just want more information about them in general. Dr. Myers was previously a professor at Georgetown University and also worked at the National Institute of Health, so knows plenty about what's going on behind the scenes here, and hoping that he'll be able to shed some light, again, things that we don't normally hear. He's also a physician policy leader, so for those of us in the pro-life movement, a fellow activist there. If you have questions for the question and answer period, which will be at approximately 12:20, you can submit those questions to Abigail, ABIGAIL, at ichooselife.org.
Hello. So thank you for joining us.
They can access your slides.
Okay.
So you've got them there,
Okay.
and they can access them.
So I'm going to sort of follow along the slides that are on the website. And the purpose today is just to talk about the science, sort of the ABCs of the vaccine. As Abigail said, the legacy media doesn't always give the full context. And so we're going to just try to explain what is an mRNA vaccine, for example. And to be clear, my purpose is not to endorse any vaccine, but just to give a scientific perspective on the process. As you know, this all began with Operation Warp Speed in April of this year, which was a somewhat innovative approach to public interagency and private pharmaceutical company, for example, partnership. And the goal of this project was to serve sort of like Homeland Security served after 9/11, to coordinate all the efforts so that the right hand would know what the left hand is doing, so to speak. So the key players in the process include the National Institute of Health, which is the central hub of federal research, and that is centered in Bethesda, Maryland. And of course, the Center for Disease Control is in Atlanta. The pharmaceutical companies are obviously involved in the developmental process of the vaccine. And then, of course, there is the policy dynamic from Congress and the executive branch of government. And Congress allocated nearly $10 billion for the project to get it going in May. So the key players right now in the vaccine race, so to speak, are the Pfizer biotech and Mrna, which used the so-called mRNA vaccine, which I'm going to explain in just a second. And then the AstraZeneca Oxford University collaboration, and also the Johnson & Johnson with Janssen Pharmaceutical, which are also using a sort of classic method of the vaccine. I'm going to explain that in just a second. As you know, the FDA is actually meeting as we speak to discuss the emergency use authorization to approve the Pfizer vaccine, which will begin distribution as soon as tomorrow and should be available by next week if it is approved, which it likely will be. So again, if you just refer to the slides, I show here a picture of the antibody against the COVID-19 spike protein. Spike protein is just a fancy term for the surface of the virus. It has a little spike on it. And what's interesting is with the human immune response, a person does not need to be exposed to the entire virus, as would happen with natural infection. But with a vaccine, the individual can be exposed to only a sequence of the virus spike protein, and that's enough to trigger the immune response. And so that's the goal of a vaccine, is to expose the patient to this little piece of the protein, which is enough to get those antibodies formed to provide protection against the virus. Now, what do we mean by mRNA? M is just messenger, like a mailman. Messenger RNA is coded from the DNA. The DNA, if you remember from your high school or college biology, is sort of the brains of the cell. And it codes for the mRNA, which provides the sequence or the code to then make the protein in the cell. Now, in this case, when we say protein, what we want is for our cell to make the spike protein so that antibodies can form against it. And what Pfizer and Moderna have done with the first ever messenger RNA vaccine is to introduce to the patient a little piece of this mRNA, which then naturally goes to the cells and codes for this spike protein to then trigger the antibodies to be formed. And this is a relatively straightforward process. Conceptually, I think of it as sort of a digital approach to a vaccine, which is actually more precise and more sophisticated than the older models of the vaccine. If you look at the examples I've given again in the slides, you'll see that with Pfizer and Moderna, mRNA vaccines begin with having this little piece of mRNA, messenger RNA, which codes for the spike protein. It goes to the cell, and then the cell basically shows it off, and the antibodies are formed. So that's basically the process. Again, little snippet, little piece of the mRNA goes to the cell, codes that spike protein, and once it's coded, your antibodies will form naturally in your body against that and provide protection. The study showed that this method was about 95% effective and was safe. Now, what's interesting is that these studies are closely monitored by what are called data safety and monitoring boards. And I used to be on some of these committees when I was at Georgetown and Washington, D.C. And these committees are very, very rigorous in terms of the scientific process. And again, I am not endorsing or supporting any vaccine, but generally speaking, I do think that the scientific process has integrity that is generally insulated from any political activity. And let's talk for a second about the sort of old school approach to making a vaccine that's being used by the AstraZeneca and Johnson & Johnson vaccine. These are not yet available and probably will not be so for a number of months. Again, the FDA is discussing approval for the Pfizer vaccine, which is one of the two messenger RNA versions. The AstraZeneca, the Johnson & Johnson are not yet available, and the studies have not been as strong in terms of they have not yet achieved that 95% threshold. But just to give context, these vaccines, as you can see in the slide illustration, basically just take a little sequence of a little piece of the protein, and what they do is they package it into a what's called adenovirus. That's basically just a common cold virus. Most people have been exposed to it whenever you have the sniffles or runny nose. That might be from an adenovirus, mild cold. So what they do is they take a weak, innocent adenovirus. They put these little protein pieces in it, and that adenovirus functions as a carrier to release these pieces of the spike protein into the body. And again, your antibodies form naturally. So that is sort of an older method. Those are the two methods that are the predominant ones among the leading vaccine candidates. There are other vaccines that are early in the process, and people can contact me on Facebook or contact Abigail at iChooseLife.org if they want to get a comprehensive list of all those vaccine candidates. One of the questions that has come up is about the use of human fetal cell lines, sometimes called immortalized. What do we mean by immortalized? Basically, that means that the cells are taken and grown in the lab and preserved in a way that can be basically sold and used in research or in industry indefinitely because they're preserved, they're sort of on the shelf, so to speak. And in terms of the fetal tissue lines that were originally discussed when George Bush gave a speech on the topic in 2005, some of the classic archived or old, if you will, fetal cell lines include the ones listed on the slide were HEK, K is for kidney, and PER, which is R is for retinal. And these are cell lines that have been developed by industry and also in coordination with Leiden University in the Netherlands back in the 1970s and 1980s. And these are, again, immortalized cell lines that are used in the production of the vaccine. To be clear, they are not in the vaccine, but they are used in the development process as little cellular factories which generate that adenovirus that I mentioned, which is necessary as a vehicle to carry that little protein piece and take it into the body for your own antibodies to form. So the AstraZeneca and the Johnson & Johnson, Janssen companies use these fetal cell lines that are archived, that are immortalized in the developmental process, but again, they are not in the vaccine itself. Now, if you look at the next slide, I talk about what are immortalized cell lines, just to give some context. There was a best-selling book that came out in 2010, and it's on a movie now, HBO. You can Google the trailer called The Immortal Life of Henrietta Lacks. And it's a fascinating story of how there was a patient in 1951 who had cervical cancer, and when the doctors did a biopsy, they took her cells and grew them in the lab. And this was the first time that cells were actually grown in the lab outside of the body and essentially immortalized so that they could be passed on from researcher to researcher. And what was controversial, to put it mildly, is that the family did not consent and did not receive compensation, even though multiple research projects and ultimately pharmaceutical products that generated billions of dollars in revenue were generated from it. And so this came out as a best-selling book in 2010 when a reporter basically exposed this process and basically advocated for the descendants of Ms. Lacks, that her so-called HeLa, H-E-L-A, which is short for Henrietta Lacks cells, were being used in research without them having known the whole story. And eventually, that was led to a conclusion in which the family was compensated and so on. But it creates a highly dramatic movie about how this process happened and how these cells were immortalized. And that gives some context of the use of cells in research. Now, to be clear, I'm not saying that the use of the Henrietta Lacks cells is a bad thing. And in her case, the problem was that she did not give consent and was not compensated. But again, that's just an example of how cells are immortalized, and that was subsequently applied to the fetal tissue lines in the 1970s and 80s that I mentioned. So the take-home thoughts, to just kind of summarize what I've said so far, is the messenger RNA or mRNA technology used by Pfizer and Moderna appear to be safe and effective, and they do not involve the use of fetal cell lines. This is the vaccine that should be available soon. And again, it appears to be scientifically valid and free from use of fetal cell lines in the production process. The Johnson & Johnson and AstraZeneca vaccines have not yet been established, but are still under study, and they do use the HEK and the retinal cell lines that I mentioned in their production process. And again, if you want to get a sense of what immortalized cell lines are all about, you could Google the Immortal Life of Henrietta Lacks trailer, just to give you an example. So I'm going to take some questions now. Again, those who want to send a question in can email Abigail at iChooseLife That's ABIGAIL at ichooselife.org. You can email questions. And we have one question here, which is, how can we take the vaccine that has no safety studies for long-term effects, no studies on its fertility or pregnancy, and how do we know with certainty that mRNA will not harm reproductive organs or future children? Well, I think that I would explain this vaccine as introducing antibodies in a way that is not unlike antibodies that form when somebody gets a common cold. We all get exposed to viruses. Again, every time somebody gets a cold or the flu, our body naturally makes antibodies. And so the process of developing antibodies is stimulated by a virus is a very normal and benign process that is generally not considered worrisome. And again, the process of these vaccine developments is monitored by what they call data safety and monitoring boards, which are very rigorous. And if there were any scientific reason to be concerned about long-term consequences, that should come out in the monitoring process. So while obviously we can't speak with absolute certainty about the future, the process does seem to be relatively safe and benign. And so Abigail is taking more questions now. So I'll pause for just a second for her to get those. Another thing I can mention is that, as you probably heard in the media, approximately half of the population is skeptical about receiving the vaccine. So again, the purpose of this discussion is just try to give some context of the science behind the vaccines. And again, it seems to be relatively straightforward. And I think that the Pfizer and the Mrna vaccines, which use the messenger RNA, are using a process which, from a biological perspective, makes sense. To just summarize it one more time, the vaccine delivers small pieces of messenger RNA, which is basically just a sequence that codes for the cells to make a little piece of protein, which is the spike protein of the virus. And then what happens is that is recognized by our body to create antibodies for immunity.
There's anything else you want to share?
So right now there are no other questions, but if somebody's interested in a comprehensive list of all the vaccines in development and the process being used by those vaccines, I have that list and I shared it with Abigail, so you can contact her at that same email if you want a comprehensive list of those vaccines that are in development. Again, the FDA is meeting today to review the emergency use authorization for the vaccine from Pfizer. And so it's expected that they will probably approve it, in which case that Pfizer vaccine should be available locally as soon as early next week. Obviously, that is somewhat speculative because we won't know for sure until the approval process is final. The vaccine will be made available to members of long-term care facilities and healthcare workers first, and should be available to most all people by March. So that is what we know so far. And again, you can email Abigail for more information on the vaccines. So I hope that gives some scientific context. again, please refer to the slides listed because that illustrates the concepts that I talked about. So I'm going to give it back to Abigail, and I think that we'll pause there for now.
Thanks for joining us. If you think of questions later that you would like to have addressed, you can still email them to me. Again, my email address is up on that website. Facebook, this may be appearing to you backwards. So ichooselife.org. Under our events tab, you can find the COVID vaccines discussion with Dr. Myers. And my email address is listed there, as well as the slides that he was working off of today for the lives. So thanks so much for joining us. Again, please share this with anyone who you think it would be helpful for, and feel free to hit us up with any questions that you think of later. Thanks.